This is your pre-lab video for the week of February 28th. Just a couple of announcements before we begin. I want to remind the people who are attending remotely this semester to be sure to get in touch with your TA if you haven't already to Zoom into lab. So you still have to attend the 2 lab sessions. This is going to be critically important for you to maintain close contact with your group members who are going to be doing the work in the lab while you do all of the data analysis online. So please make sure that you're attending labs so that you can stay in the loop. Will, for the rest of the semester, be a no shorts, no open-toed shoes lab. This is very common. It's for safety reasons, and I'm going to be reminding you a lot as the weather gets nicer that this isn't allowed in the lab. On Friday, we'll have the 3rd in our series of 5 weekly workshops. These are optional workshops for you covering subjects that we've identified as historically being problems for students. And we thought it would be a good idea to formally reinstate our workshop series in light of how hard it is for us to get to know each other and to find resources when most things are online. This week, we're going to be telling you about how to get more research experience. People in the Discovery Lab often leave the class really wanting or needing more research experience and are at a loss as to where to find it. So we'll help you make a plan for how you will get more research experience. We're going to invite a number of students who are involved in research in a number of different ways, not just the honors thesis or independent study students, but we hope that the students will be able to give you some resources and ideas for other ways to get research experience while you're an undergrad in MCDB. This week, you read a perusal reading on the molecular biology of radiation. We'll be covering that subject in lecture this week. And this week in lab, you're going to be starting your experiments. So what we'll be doing is isolating embryos and isolating 3rd instar larvae to start setting up your controls. And then the following week, the week of March 7th, you'll be setting up your first experiments based on what you proposed in your proposal that you submitted this past Sunday. We're walking you through the development of your research project. This week, you should have submitted your research proposal, which was through Google Forms. Excuse me. Those we're going to be taking a look at. We'll be grading them. And we'll also be distributing each of the proposals to 2 or 3 other research groups in the Discovery Lab for peer review. Now, peer review is a critical part of research where somebody who has the same level of expertise as you do takes a look at your work to see if there are gaps, if it makes sense, and if you think it's feasible. So they'll be giving you feedback also. Your completion of the peer review will be graded, and your peer reviews for other people will be distributed to them with your names taken off. So it's an anonymous peer review. So that's the second step in your research project. We did the proposal, then you have your peer review, and then we're going to start chipping away at the different components of a research poster as you're doing your research, with the idea that within about a month or so, when your research is mostly complete, you'll be putting in your data and then thinking about the discussion section where you think about the future directions of the project. And then on April 21st, I think it's the 21st, Wednesday the 21st, we're going to have the Cure Symposium. It will be held as a remote symposium, much like all of the national meetings. And we're going to help you understand how to give a great presentation at a symposium to present your research at the end of the semester. Now, we'll be giving you your proposals to peer review at the beginning of the week, so you can work on that in the first lab. And I recommend doing that in lab so that you can work with your group members. You'll just be submitting one form, one evaluation for all of the proposals that you receive. So you'll be completing it as a group and just submitting one form. So do that in the first, I think you should do that in the first lab just to get it done. And then you can focus on the techniques that you'll be learning this week in preparation for doing your experiment next week. Now, this past week, you set up population cages. We put a lot of flies into containers with grape juice agar plates and yeast paste and lots of space, and they've been breeding like crazy. They started off slow, but today is Sunday, the 28th, and I was in there today isolating embryos, and we have a ton of embryos, and that's excellent because those are the embryos that become the third instar larvae that are your model organism for your compound. So we're going to be maintaining those same cages for as long as we can to try to get as many embryos out of those flies as possible. This week, in the first lab, you are going to be taking the grape juice plates in the cages in the lab. You'll be changing them out, so each section will have one or two of those plates to isolate embryos from. So let's take a look at the protocol. These are videos that Gretchen made that hopefully will show you exactly what you need to do before you come to the lab and before your TA explains it. So what she's doing is she has opened up the top of the cage. So you did this last week when you were feeding the cages, and we're going to continue doing that every single day. We're going to be putting a new plate in there. One of the tricks to not getting the flies to come out is to tap the top, because remember that flies really only know up and they know light. So they really like to hang out on the inside of this cage on the top. So if you tap it with your fingers, they'll fall down. And then when you open up the top, they won't come flying out the hole. So she's going to reach inside and she's going to take that plate like you've been doing when you've been feeding, tap off all of the live flies. And you can see that she's tapping it quite a lot to try to get as many flies off of that plate as possible. And she pulls it through the hole and immediately recloses it to prevent the flies from coming out while she's isolating the embryos. You can worry about replacing the plate afterwards. Now, when you look at the plate, if you didn't do this last week, take a really close look at it this week. What you see is the grape juice agar, also the yeast paste. You can still see that there. But you can also see these little white dots, and the little white dots are all embryos. Females lay between 150 and 300 embryos at a time. So there are many, many, many embryos on this plate. Many of them will be fertilized, some won't, and those are just eggs. We just call everything an embryo because the vast majority of the eggs on the plate are fertilized. You're also going to see on the side some black dots that are even smaller than the embryos. These are poop. So don't isolate the poop. That'll generally just stay stuck, and I'll explain in a second why we don't really worry about it that much. But the embryos, for the most part, are all inside where you put the yeast paste. So to get the eggs out of the yeast paste, you need to make the yeast paste into a slurry. And the way that you do that is by adding water. So Gretchen here is spraying it, and what she has here is called an egg collection basket at the bottom. She has that inside of a container. That egg collection basket they're just craft containers. I sawed off the bottom and then sewed veiling on the bottom because the size of veiling for weddings is actually perfect for collecting embryos. So if you make the yeast paste into a liquid and allow it to flow through the veiling's going to trap the embryos on the top and allow all of the liquid yeast paste to go through. You can also, instead of spraying the surface, you can actually just put the plate on the surface of the bench and pour a little bit of water on top. And then what you'll be doing in either case is you'll be loosening up the eggs with a paintbrush. So you can see that Gretchen's doing that here. She's loosening up the yeast paste because it's nice and wet. And she's going to continue to wet it and drain that yeast paste through the egg collection basket into the container, which will collect the liquid. Now you can see that in here there are a couple of dead flies. There are also all of the embryos here, nice and clean and ready to go into new food. So she continues to wash it off. And in the end, when you're done, you should have a perfectly clean plate. Now what's kind of cool here is you can see in the middle, you'll be able to see this even better in the lab, that it's not purple anymore in the middle because the flies love that yeast paste. And they also drink the grape juice out of that agar, which creates that sort of jello substance. And so you can see where they've been eating and they've been laying the eggs right on top of the yeast paste. So she also runs that extra liquid through one more time because sometimes you can get a lot of extra embryos out of it. And you can see that there are a few more there. So then she takes the embryos and she's going to pick them up with a paintbrush. I think that she's probably trying to avoid the dead flies. You can just take those out, or you can just try to avoid them as you're picking it up. She's taps the embryos into the bottle. And then what you can do, which is nice for the flies, for the developing embryos, is to take a wet paintbrush and just kind of paint them along the surface of the food. So she's going in and she's taking as many as she can out of this egg collection basket. In your case, because this video was made really early on, your egg collection basket is going to be loaded with embryos. You're going to have probably about 50 times what she had in there, because that was when the population cages were very first set up. So they're just kind of getting used to the setting. So we're going to allow those embryos to hang out in the food and develop for about 5 days, at which point they'll become third instar larvae. So if you remember from your fly development lesson, they hatch, they become first instar larvae, second instar larvae, third instar larvae. That's the largest larva that you'll see before they become pupae, or the cocoons that you also can see on the sides of the bottles. So to isolate the third instar larvae, it's really important that we have only third instar larvae. The plates are collected every 24 hours. So the age range is anywhere from 0 to 24 hours of age. And in fly life, it's a pretty significant difference. So what she's doing here is she's running some water into the bottles. Ideally, you want the temperature of the water to be kind of lukewarm. You don't want to touch it and be like, whoa, that's hot, or, oh, that's cold. Somewhere in between, something that would be almost sort of room temperature. And she's swishing it around and then dumping the food and the larvae, all of the contents of those bottles, onto the top tier of 3 sieves. So the 3 sieves each have a different size hole. So you'll see it looks like screen. And the third instar larvae are going to get trapped in the middle sieve. She's washing through the food and the larvae. The biggest holes are on the top, the middle holes are in the middle, and the tiniest holes are on the bottom. And what she wants is all of the food and the larvae to go through. You'll get bigger chunks on the top, and she's breaking them apart with her fingers. You might think, oh my God, she's not wearing gloves. She's not wearing gloves. We recommend that you not wear gloves because you're going to be able to feel the larvae a lot better if you're not wearing gloves. If you are wearing gloves, there's a high probability that you're going to end up just squishing them. And if you squish them and they die before they ever end up in your drug and food combination or get irradiated, then your whole experiment's going to fail. So you'll see when you take the tiers apart, the middle sieve is going to contain the third instar larvae, and then the bottom tier will contain the smallest larvae and some additional eggs. So you can wash that smallest tier, the lowest level, so that all of those tiny larvae end up in a pile. And at this point, your TA will tell you what you should do. You can either take them and scoop them into bottles, about that size clump, so that you have somewhere in the range of 250 to 500 larvae in each bottle, or you can just put them into the freezer. This will kill them, and then we can autoclave them. It takes about 15 minutes for them to die in the freezer. Do not freeze the middle tier. That's the one that contains the third instar larvae. Gretchen is washing the third instar larvae down into a pile, and this will allow her to scoop them into the irradiation container, which is just a plastic box. That's the thing that's going to go into the radiation machine to irradiate the third instar larvae. So very carefully, she's going to take the metal spatula and she's going to scoop them into that container, and she's going to cover them with yet another form of pantyhose. So that's what will go into the radiation machine. When those larvae come out of the radiation machine, they're going to be ready to transfer into your vials. Now the vials we have to make. So what you're going to do is you're going to make food, or there will be food made for you, and you're going to transfer 5 milliliters of food plus your compound, which in the first week, this coming week, will be all controls. You'll be either using colchicine or you'll be using a negative control. And we're going to assign each of the sections different negative controls: DMSO, methanol, ethanol, or water, because these are the common solvents that are going to be used in Discovery Lab. The reason we're doing controls first is twofold. First, we want you to get some experience in a sort of low stakes environment. You'll know right away if your vials don't look good. And two, having everybody work on controls in the first week is going to give us an opportunity to have a ton of controls and have more statistical power going into your experiments next week. To set up the vials, you'll have food. Now, all of those food bottles in the lab, we make that food, and you'll be doing the same thing in the coming weeks. The food is liquid. It has agar in it, just like the grape juice plates, which will solidify in a vial or in a bottle. You can transfer the food by measuring it into a large syringe and squeezing it in so that you have 5 milliliters into each of the vials. Another thing that you can do is you can measure 5 milliliters either with a graduated cylinder or with your P1000. You can measure 5 milliliters, 5 1,000 microliter volumes, and mark it on the side of your vials. This visually can be really a nice way of getting the food in there, and also it prevents you from having to reload the syringe every single time you set up a vial, and you're going to be setting up quite a few. Gretchen's pouring it here. I can tell you that that's very, very hard. I actually load the entire syringe with a lot of food, and then just line up the vials and put 5 milliliters, 5 milliliters, 5 milliliters as I go down the row. Here she's putting the controls in. You're going to have tubes that contain either the negative control or the positive control, and you're going to put 5 microliters into the food. Do not pipe it up and down, and mix around just with the tip. It will diffuse through the food as the food is solidifying so that the larvae will eat the same amount. You'll also want to label the vials either negative or positive before setting them aside, because if they're not labeled and we find them in the lab, they're going in the trash. After the food is solidified and the larvae have returned from being irradiated, you'll be putting what we call a blop of larvae on the top surface of the food, and you should aim to get somewhere between 50 and 100 larvae. This isn't going to be easy to estimate, but we will have an idea next week of how close we were to that estimate, and you'll get better at it. So just do the best you can in the first week to put a blob of larvae that represents what you think is 50 to 100. Fewer than 10 larvae in the vial we can quantify it, but it won't be statistically powerful enough. So we need to have enough larvae in the vial so that when we count it, we know that it's statistically meaningful. So this week, again, you're going to be working on peer review. You'll be isolating embryos. In the second class, you'll be isolating embryos, the third instar larvae, and putting them after irradiation into the food drug vials that contain positive or negative controls. So your peer reviews are due on Sunday, March 7th, as is the Next Play Posit, which is on Tintin Sioux Labs research. And then you'll be setting up next week, the week of March 7th, your compound vials. So our hope is that all of the compounds that have been ordered will be in, and you'll begin your experiments then. You'll be spending the next 3 weeks setting up compound vials, and during that time, the very first vials will start to a close, and you'll be quantifying them. So in the next month, you'll be in a rhythm of coming into the lab, feeding the cages, isolating embryos, isolating third instar, setting up vials. So that's kind of what it's going to look like for the next month. That iteration is going to give you a lot of really great experience and hopefully enough vials to get a really fantastic answer for your compound.